Hi everyone. Glycolysis pathway tends to have several pathways which may be very confusing for the students, and some of them always ask, how do I understand all these steps? So in most cases, some students take the path of trying to cram everything, but it's not usually the most advisable path to follow, because it's easier if you understand. Once you understand, recalling the processes, it's easier. So in this video, we'll have a simplified explanation of what glycolysis is, and with that you'll be able to understand each and every step, which will make it easier to remember and even memorize the enzymes involved. So first thing first, the first question is, what is glycolysis? This is the breakdown of glucose to form pyruvate, and in most cases it's usually for production of energy, and it usually facilitates both the aerobic and anaerobic cellular respiration. So basically, glycolysis has 10 steps, and it's divided into two major phases. So step 1 to step 5 covers the first phase, and then step 2 to step 6 to step 10 covers the second phase. So the first phase involves consumption of 2 ATPs, while the second step involves the production of 4 ATPs. So the first step in glycolysis is the conversion of the glucose to glucose 6-phosphate, and the process is catalyzed by the hexokinase enzyme. So as you can see here, we have our glucose, which is the 6-membered carbon, and here is the ATP, and of course in this case it's consumed, and we have our hexokinase here, and it uses magnesium as a cofactor. So in the final product, we have the one of the phosphate groups will move from ATP to glucose, so that it can form the glucose 6-phosphate, and of course the ATP will now be converted to ADP, and there will also be liberation of the hydrogen that was initially here. Okay, the second step involved in glycolysis is the conversion of glucose 6-phosphate to fructose 6-phosphate, and the process is mediated by the enzyme called phosphoglucose isomerase. So basically, the process involves rearrangement of glucose 6-phosphate, as we can see in this diagram here. So basically, we had our phosphate here, and it will now move to the other side. So in step 3, the fructose 6-phosphate formed will be converted to the fructose 1,6-biphosphate. This will be through another consumption of ATP, as you can see from this diagram here, where we have our fructose 6-phosphate. Then of course 1 ATP will be consumed, and the enzyme involved is called phosphofructokinase. So the kinase part involves enzymes that mediates the phosphorylation processes. So here we'll end up with fructose 1,6-biphosphate. As you can see in this diagram here, the phosphate groups are attached to the first and 6 carbon, hence the name fructose 1,6-biphosphate. So bi stands for the 2 phosphates. So in the next step here, we have the aldolase enzyme will split the fructose 1,6-biphosphate that was formed in step 3 to form 2 sugars. And the sugars in this case will be dihydroxyacetone phosphate and diglyceraldehyde 3-phosphate. So here we have the structures of the 2 sugars that are going to be formed, and the isomers of each other. So we'll have this and this. And then of course here we have triphosphate isomerase, which now helps in the interconversion of one to the other. So in the 5th step, we have the two sugars that were formed, remember they're isomers of each other, so we have dihydroxyacetone phosphate, and here we have glyceraldehyde 3-phosphate. So as we had mentioned initially, so we have the enzyme triose phosphate isomerase, which helps in the interconversion of the other. And because as we'll observe in step 6, this one here will be used up in step 6, so it means the equilibrium will shift towards this side. So a lot of glyceraldehyde 3-phosphate will be formed from dihydroxyacetone. Yeah. So in step 6, as we had said before, glyceraldehyde 3-phosphate that is formed in step 5 will be used to form 1,3-biphosphogycerate. So for the biphosphogycerate, we have 2 phosphate groups, but remember in this case they are free phosphate groups. They are not those because the processes that we had initially, it's the ADP that was being cleaved to form ADP, so that the phosphate groups can react. But in this case, we have glyceraldehyde 3-phosphate that will react with free phosphate group to form 1,3-biphosphogycerate. In step 7, the 1,3-biphosphogycerate formed in step 6 will be converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. As you can see from the diagram here we have the 1,3-biphosphogycerate that was formed in step 6, and then here the 2 ADP molecules will react to the phosphate groups, so that in the end we'll have 3-phosphoglycerate and 2 ATP molecules. So in step 8, the phosphoglycerate formed in step 7 will be converted to 2-phosphoglycerate. If you notice, it's the positioning of the phosphate group that will change in this case, and the enzyme involved will be phosphoglycerate mutase, which will now help in the interconversion or the rearrangement of this enzyme, so that it can come from the 3rd carbon, the phosphate group can move from the 3rd carbon to the 2nd carbon. In step 9, the enzyme enolase will be involved in removing water molecule that is from the 2-phosphoglycerate, so that it can form phosphoenol pyruvate, as you can see here. So the water will be removed here, so that in the end we'll have the phosphoenol pyruvate. Yeah. So in the final step, the phosphoenol pyruvate will be converted to pyruvate, and the process will also involve formation of 2 ATP molecules. Which means the process helps in the liberation of energy. Remember, this energy will be used up in the processes that follow glycolysis. So we have like the Krebs cycle. So in summary, here we have now the overall process of, we have all the steps involved. As I had said initially, we have 10 steps that are involved. So in the 10 steps, we have 2 phases. Phase 1 is from step 1 to step 5, and then step 2 is phase 2 is from 6 to 10. So in the first phase, we had, as I had said initially, glucose was converted to glucose 6-phosphate by hexokinase, and then glucose 6-phosphate was rearranged to form fructose 6-phosphate, and the enzyme involved was phosphoglucose isomerase, and then there was now the first ATP that was consumed for glucose 6-phosphate to form fructose 1,6-biphosphate. And then here we have the enzyme aldolase, which cleaved the fructose 1,6-biphosphate to form the hydroxyacetone and glyceraldehyde 3-phosphate. And then also remember we have the triose phosphate isomerase, which now converts the dihydroxyacetone phosphate to glyceraldehyde, which is now used up in the next step, which is now step 6. Yeah, and of course now in step 6 there is formation of here we remember we had talked of a free phosphate group reacting. The phosphate group here is not from ATP, so maybe that's one thing we'll need to remember. So the reaction will lead to the formation of 1,3-biphosphoglycerate. So the 1,3-biphosphoglycerate formed here will again lead to the formation of now ATP, because here we can see there is ADP, which is going to ATP, and then with that which will lead to the formation of 3-phosphoglycerate, and then the 3-phosphoglycerate will undergo rearrangement in step 8 to form 2-phosphoglycerate, and then the 2-phosphoglycerate will be dehydrated by the enolase enzyme to form phosphoenol pyruvate, and then finally the phosphoenol pyruvate will be cleaved. Now the phosphate group will react with ADP to form ATP, and finally we'll have pyruvate, and in this process here also ATP will be synthesized or will be formed. So overall the consumption of ATP in glycolysis, you have 2 ATPs that are consumed and 4 ATPs that are produced in this other process. The overall production of ATP is 2. Since 4 will be formed but 2 are consumed, the process consumes 2. So definitely at the end of the day we'll have 2. So the process is quite simple. You don't have to cram. All you have to do is to understand what happens in each process. That's why it would be easier to remember the enzyme. For example, if you the term hexokinase, you'll definitely know. For the kinase part, is involved in some form of phosphorylation. If it's isomerase, of course it has to do with formation of isomers. If it's phosphofructokinase, of course the kinase part means there's phosphorylation. So the overall glycolysis equation is as shown. So here we have the glucose, which, as we had said before, it contains 6 carbon atoms. Then it will react with 2 NAD and 2 ATP and 2 phosphate groups to form 2 pyruvic acids, 2 ATP, 2 NADH, and 2 hydrogen ions. So thank you very much for listening. If you have any questions or comments, don't hesitate to write on the comment section. If you need any form of tutoring, that is a topic in biochemistry, you can also contact me through email. I do it at a subsidized price. So thank you very much for listening and have a good day.